Vermont. And I'm really happy to welcome you all on behalf of UVM and our partners, Tufts University and Harvard University. And a really big thank you to the folks at the Wyss Institute for Biologically Inspired Engineering at Harvard for organizing and hosting this conversation. We're really excited about it. So we're here to talk together about Xenobots. Some people refer to them as Xenobots. We can have a separate conversation about that sometime. These are the world's first living robots, and they are, in fact, I would argue, something new under the sun. They tap into, help us understand ancient forces as old as life itself. These are hybrids, biological machines that represent an amazing synthesis between computer design, human insight, laboratory expertise, artificial intelligence, and the innate capabilities and mechanisms of cells. Reported last year, Xenobots are computer designed. They're hand-built biological machines. They're assembled from frog cells that have been shown to be able to move on their own, find targets, retain information. And now, as you may know, in a new study published this week in PNAS, the Proceedings of the National Academy of Sciences, the science team, the folks gathered here today, report that Xenobots are able to self-replicate. It's a form of biological reproduction never seen before. So let me introduce you to the team. Josh Bongard is the Wynot Professor of Computer Science here at the University of Vermont and Director of the Morphology, Evolution, and Cognition Laboratory within UVM's College of Engineering and Mathematical Sciences. Mike Levin is the Vannevar Professor within the Department of Biology at Tufts University and the Director of the Allen Discovery Center at Tufts and the Center for Regenerative and Developmental Biology at Tufts. Mike is also a distinguished professor and associate faculty in Harvard's Wyss Institute. Sam Kriegman is a computer scientist who completed his doctoral work with Josh Bongard here at UVM and now holds a joint postdoctoral appointment at the Wyss Institute and at the Allen Discovery Center at Tufts. And Douglas Blackiston. Doug is the senior scientist at the Allen Discovery Center at Tufts University and at the Wyss Institute at Harvard as well. So it's now my pleasure to turn this over to my colleague, Professor Kirk Dombrowski. Kirk is the Vice President for Research here at UVM and a distinguished anthropologist and scientist in his own right, and a key leader and driving force behind the work that you're going to learn about during this webinar. So Kirk, over to you.
Thank you, Josh. I'm delighted to welcome you to this webinar and to this conversation about a remarkable discovery and a remarkable new chapter in science. Like many of you on the webinar today, I'm amazed at this discovery, or should I call it invention, of living robots, and now this new result that they can replicate. It's a new form of biological reproduction. The scientists who made this discovery, who invented Xenobots, are here, and I'm going to turn this over to them shortly, but I want to take a minute and give you a sense of the partnership and larger landscape that's led us here. This ongoing research endeavor is built on partnership, a partnership that's allowed a team of researchers to create and understand these Xenobots, and which combines computer science and biology in truly novel ways. The partnership began as a collaboration between the University of Vermont and Tufts University and led to the creation in late 2000 of the Institute for Computationally Designed Organisms, the ICDO. I'd urge you to take a look at the ICDO website to learn more. The ICDO spans two locations, the Allen Discovery Center at Tufts University, led by Michael Levin, and the Morphology, Evolution, and Cognition Laboratory at the University of Vermont, led by Josh Bongard. And I'm delighted that this partnership is growing and deepening and is now expanded to include our wonderful host today at the Wyss Institute for Biologically Inspired Engineering at Harvard University. Please consider this a special thank you to our new partners. The most recent news and reason for being here today is the newest capacity of Xenobots, self-replication, the discovery that the world's first living computer-designed organisms can perform novel forms of replication. a challenging and potentially game-changing discovery that the researchers will discuss more fully. As importantly, though, the larger goal of the research and the Institute is a deeper understanding of life itself. Xenobots, the science team has said, are tools for learning. And in this time of rapid change and multiplying global challenges, from climate change to burgeoning pollution, we have much to learn. The intersection and interaction of life, machines, and intelligence bends our categories and opens important opportunities, from regenerative medicine to environmental cleanup to new insights about how life works. And Xenobots themselves present new challenges and new questions. This partnership, the team's approach, and the deep quest for expanding life science will help researchers around the world to advance innovation at the frontier of science and technology, and to address the vitally important ethical and philosophical questions inherent in this new work. I look forward to learning more in the conversation that follows, and I'm happy to introduce Lindsey Brownell, a science writer and communicator at the Wyss Institute, who will serve as the moderator for this afternoon's conversation. Lindsey.
Thank you so much, Kirk, for those wonderful opening remarks. And again, welcome to all of our attendees. If you would like to see a transcript of this event today, you can go to the bottom of your Zoom screen and click More, and then click Show Subtitle. We want to make sure that everybody can access this information in a way that is best for them. So we are going to be having a really fun discussion today with all of our scientists who are the authors of the Xenobots 3.0, as we're calling them, paper. And it has created quite a stir on social media over the last few days. So I'm really excited that we can sort of respond to some of these questions and comments that people have been making about them, because as Josh Brown said earlier, they really are something new under the sun. So I'm going to start by asking my first question to Mike Levin. So for anybody in the audience who may not have been following the trajectory of Xenobots, could you just explain what Xenobots are? What are these little blobs that we've seen tootling around in a dish?
Sure. Xenobots are a new entry in a long tradition of experiments designed to understand a number of questions in different fields. So we are still, as scientists, groping for a proper understanding of what robots are. What does it mean to be a robot? What does it mean to be an individual? What does it mean to be an organism or a machine? And this is part of our effort to understand biological plasticity as well. You know, what are collections of cells able to do under various circumstances? And so what Xenobots are an extremely convenient model system to ask questions like this. Basically, they consist of skin cells taken off of an early frog embryo. In one version, there's also a bit of muscle put in as well, but they are basically taken off or liberated from the normal instructive interactions that tell them what to do as skin, you know, to sit on the outside of a tadpole and keep out the bacteria and so on. And in this new environment, right, they're placed in a new environment. They're stimulated or sculpted in accordance with the instructions of a machine learning algorithm, about which you'll hear more today. And then they're given the opportunity to basically reboot their multicellularity. You know, they're as a collection of skin cells, they could have done many things. They could have made a monolayer or crawled off, or, you know, but what they in fact do is form a very coherent little proto-organism that has some surprising capacities. So it's motile, it's self-actuated, it can move, it can heal damage, as we found out and reported this week. It can actually replicate and make, under very specific conditions, make copies of itself, and many other things which we're only now beginning to sort of scratch the surface of what they can do. But it's an example of amazing biological plasticity.
Awesome, thank you. So my next question is for Doug, whom I know was in the lab doing a lot of the hands-on work to create these Xenobots. And as I mentioned, a lot of people since the paper came out have been very concerned about what these things could or could not do now that they are living robots that can replicate. And a lot of people have been referring to living robots in sci-fi movies and TV shows like the Cylons from Battlestar Galactica, which famously are robots, but they are biological. They look like humans and they end up terrorizing the human race and basically committing genocide, which obviously nobody wants to see happen. So as the person who was most closely involved with building the Xenobots and seeing what they can and cannot do. Can you just let us know what the process was for actually, coaxing these skin cells to do this other stuff that they can now do, as Mike said, liberated from their normal environment of being, part of an embryo. And then can you let us know what they can and cannot do? How much should we actually be concerned about?
Sure, thanks so much. Maybe I'll walk you through the design process for just 1 or 2 minutes, very briefly, and then we can get into the ethics. So in general, you can think of these like small microscopic self-motile cellular forms or designs that are all built from amphibian cells. And we started here with amphibians because there's a lot of advantages, although we could easily port this to other cell types as well. So amphibians live in water, so that's convenient for us. You can keep them on your benchtop and in fresh water at room temperatures, and these cells survive, and these forms survive for over 10 to 14 days without any external food source because they're loaded with energy from the egg. Also, there's a rich history of using these cells in developmental biology. So for over 100 years, this has been a laboratory species. It was established by a rather eccentric scientist named Lancelot Hogben, who was a British zoologist from South Africa and brought the species back. Since then, the field has flourished and this model has been used. So we have a nice atlas of where different cell types and stem cells can be collected from throughout development. And there's been a lot of modern work looking at the molecular biology of cell fate specification, and we can play with the molecular genetics to get different cell types. So what I do is I have a couple different design methods. In one, I harvest individual cells or tissues from different parts of the embryo to build something new from the ground up. So we did this in our original work, where we placed and layered cardiac cells to produce a specific type of walking motion. But we can also take whole regions, like a sphere of tissue of the skin that people have studied for many decades, that develop cilia. And these cilia beating little hairs create motion. And we did that in a manuscript as well. And for both, we can shape these and push them into different geometries and anatomies using a series of tools. And then finally, some of the other team members will talk about the fact that we can also simulate these designs. So we can use a computer simulation in a virtual world to explore the types of behaviors or interesting phenomenon that we might want to generate. So regardless of the design, I often think of all of these as just different living materials with which we build. You could think of them as actuators for movement, scaffolds, and sensors. So after the design aspect, there's certainly ethics. I just want to specifically say that this is something we're very sensitive to as scientists, and this falls under multiple levels of oversight. So there's institutional regulations that are well established for anything involving living or animal material, as well as state and federal regulations, which have been worked out for many microbial systems that are genetically modified in laboratories. Also, I want to say at the university level, we have a committee that oversees any of our animal work. And this contains not only veterinarians and scientists, but also upstanding members of the community, which include clergy, lawyers, and ethicists that have to make sure that this passes the general ethics guidelines. And last, as you hinted to earlier, this system is extremely contrived, and what we see with the self-replication that some of the other members will talk about, it really only exists under a very specific set of conditions in a petri dish right now. So without supplying a rich source of frog stem cells to the system that are added externally by an investigator, the system falls apart and essentially the cells disintegrate after 10 to 14 days. So this is not capable of propagating without outside help.
So if these Xenobots were to, escape the lab and end up in a pond somewhere, eventually, within about 2 weeks, there just wouldn't be any more of them.
Sure. So frogs shed their skin all the time. When they shed their skin, there's often healthy cells, of which it's the same cell types we're using to build these constructs. And those cells go on to just decompose in the waterway and are harmless.
Excellent. That's very reassuring, actually. So my next question is going to be for Josh, because this collaboration, as we've just been talking about, is a lot of developmental biology has informed this, but there's also a computational and computer science aspect to it. And so, Josh, my question for you is about the fact that we call these things living robots, but the Xenobots don't really have brains or any neural tissue to speak of. They're just skin cells and maybe occasionally some muscle cells in there. But they do seem to have some kind of basal cognition, because watching the videos that have been posted online, you can see that they're able to kind of navigate the environment. They're able to collect these loose cells and form them into sort of what we call the baby Xenobots that eventually become the offspring. So even though there's no brain involved, there's still some kind of cognitive function. So what can building things like Xenobots tell us about what intelligence and thinking actually are?
Yeah, that's a great question, Lindsey. Thank you. So I'll start with brains. You mentioned brains. The Xenobots don't have brains. Looking at the Xenobots through the lens of AI and robotics, if you look at how AI has developed, we've sort of said to ourselves, well, humans, we have big brains and we're smart, at least some of the time. So it seems obvious that brains must equal intelligence. And this is one of the reasons why AI researchers are building ever larger electronic brains called neural networks or deep learners. We're dropping them into phones and self-driving cars. But all of us, I think, are starting to see the limitations of this AI approach, from funny autocorrect fails to the unintentional killing of pedestrians by self-driving cars. It's not that AI does this on purpose. All current AI are solipsists. AI simply doesn't know that any one or anything else exists. On the flip side, organisms, including those without brains, like xenobots, use their bodies to push against the world and against other creatures, literally and figuratively, and they learn how those things push back. This allows them to figure out how to get what they need by working with others rather than ignoring them. And so this suggests, what we're learning from xenobots and other synthetic biology attempts, is that it suggests that intelligence is not something stored somewhere in the brain. Whatever is going on is a much more complex dance between body, brain, environment, and other sentient beings.
Wow, that's actually very profound for us as humans too, because we like to think that we are, individual thinking, self-aware beings. But yeah, we are part of our environment as well, and our brain does send inputs out, but it also takes lots of things in when trying to figure out what is up and what is down.
Absolutely.
Thank you. My next question is for Sam, who is also a little bit more on the computational side of things, and I want to focus on the replication, that aspect of the xenobots, which is, that's the new big thing that they can do. They can replicate, but the type of replication that they are doing is something that has never really been seen in biological creatures before. And as I'm sure most of us know, animals, plants, amoebas, fungi usually reproduce in one of a few ways. There's sexual or asexual reproduction. A lot of things reproduce by just budding parts of themselves off that then become new offspring. But in all of these existing types of biological reproduction, there is a component of the parent that ends up going into the child, just physically, whether it's just DNA or whether it's, there's parts of the mother and the father that make the baby. But in this case, the xenobots were collecting all these loose stem cells that were separate from themselves and were kind of aggregating them together. And then that became a new xenobot, which is sort of a replication by assembly, almost kind of like building things out of Lego blocks. So what are the implications of seeing this kind of replication in living organisms for the first time?
Well, the xenobots tend to raise more questions than they answer. So I'm not sure exactly what the implications are at this point. If we zoom in on a xenobot to the level of a single cell, it's not new. It's a part of a frog. But then we zoom back out, and a xenobot doesn't look like or replicate like anything we've ever seen before in nature. They look more like robots building other robots, as you mentioned, more like robots than an organism reproducing. And in fact, around the same time that the very first autonomous robots were being built in the late 1940s, scientists were thinking about whether or not it would be possible for a machine to build a copy of itself. And it wasn't at all obvious that this would be possible. Many thought that it would result in a logical contradiction, because it wasn't really clear whether or not an infinite regress of blueprints inside of blueprints for the entire, possibly infinite family tree would need to be somehow stored inside of each self-reproducing machine. But in 1948, John von Neumann proved that it was indeed theoretically possible to create machines that reproduce themselves mechanically. And inspired by this, engineers have since hand-designed wooden blocks with complementary hooks and levers, robotic trains on tracks, as well as robot arms that can be set up to yield copies of themselves. And the Xenobots now demonstrate something pretty cool, in my opinion, that so-called von Neumann machines can be built out of biological cells instead of synthetic materials like wood or plastic or metal, and that they can be automatically designed and optimized.
Cool. I think that that's a really big shift in that we are seeing in people's conceptions of robots. I know at the beginning, Mike mentioned that people have been trying to figure out what do we consider a robot to be. And I think for a long time, a robot was something that was mechanical, that was built out of gears and sprockets, and that was, totally separate from things that are created, but I would say naturally or biologically. But now we're seeing, biological things that can also be materials, and we can learn how to manipulate them and take advantage of a lot of their abilities that they have that, other non-living materials don't have. And that actually kind of segues nicely to my next question, which is going back to Doug. One thing that I want to call out about these Xenobots is that their genes were not altered at all. And that these behaviors that we saw them doing that were not what we would expect from a normal skin cell being part of a frog all happened without touching their genomes. And I think a lot of people, when we talk about biological things being modified or having different behaviors, a lot of people think about things like CRISPR and gene engineering. But this kind of implies that, similar to what Josh was saying about how intelligence doesn't just live in the brain, we're seeing that behavior also isn't something that lives in the genes. It seems to arise from other places. So if these behaviors weren't coded into the frog cell's DNA, where did they come from? What caused them to manifest like that?
Yeah, so I think there's a lot of developmental biology that goes into this, with the way that the cells will communicate and self-assemble and decision make as they're placed into these different contexts. And so there are some unexpected interactions that can happen there. And then what's interesting is also, on the flip side, there are some of these behaviors that are predictable. So when a cilia beats, you can generate flow, which may generate motility. And when muscle contracts, you can deform tissue in a way that might generate a walking behavior. So if you think about these things like springs and scaffolds and sensors, in addition to cells, I think the question then is how do you order the parts or put them together or allow them to assemble in a way to learn about or to perform or to enhance any specific behavior that you're interested in? So a lot of this falls in the developmental biology, but a lot of this also comes back to the simulator. So the simulator can survey this incredibly wide design space to allow us to hone in on interesting shapes, morphologies, and forms that can generate really interesting hypotheses about the types of behaviors that we may be able to generate or interrogate with the system.
Awesome, cool, thank you. And you mentioned the simulator, which is the topic of my next question, which is going back to Sam. I think that when a lot of people saw in the information about Xenobots that it was AI designed, I think a lot of people sort of assumed that the AI was built into the Xenobots, and that the Xenobots were somehow using some kind of artificial intelligence to do what they were doing. But the AI part is actually, it was instrumental in helping to create the Xenobots, but it's not part of the organisms themselves. So could you just explain how that simulator was used to help you design the Xenobots?
Sure. So here in this project, AI is a means to an end, which is a Xenobot that does what we want it to do. In more generally, the utility of AI as a design tool is that it speeds the design process, and it can come up with a diversity of unique designs that a human engineer might not think of. So in this project, we discovered something interesting, a unique form of replication. But there was a problem. This phenomenon was somewhat ephemeral. Self-replication only lasted for 1 or 2 generations. So we created a computer program that was rather simple. And all it did was predict how different Xenobot designs could prolong this behavior of self-replication that we observed. We know that body shape affects behavior. So we tried out billions of different Xenobot shapes in this simulation. And one of the best shapes was, funny enough, reminiscent of Pac-Man, a circular shape with a mouth. And at first glance, this was rather surprising. But in hindsight, after watching the behaviors in simulation and in the real living Xenobots, we have a much better intuition about why this computer design is so powerful. The mouth is used by the Xenobots to capture, collect, and essentially shovel loose cells into much larger piles than other designs could achieve. And because larger piles in this case equals bigger and stronger offspring, which are more likely to replicate themselves, it amplified self-replication.
Gotcha. Thank you. It's so cool that we have the ability to try out billions of shapes without having to do billions of experiments. I'm sure that Doug was very happy about that. Okay, my next two questions are going to be zooming back out a little bit. We've been talking a lot about the individual Xenobots themselves, and now we're going to be taking Xenobots in kind of a larger context. Mike, I'm going to put this one to you. In an earlier conversation that we had about Xenobots before the webinar today, Mike, you said that they force us to acknowledge the non-binary nature of science, as in Xenobots are not fully what we would consider a normal living organism. As Josh pointed out, they don't have brains. They can replicate only for a few generations and then they die out. But they're also certainly alive in that they're composed of living frog cells that are complete, intact, living cells that match our definition of what a cell needs to be alive. And I thought that was a very interesting statement because your background is in computer science, which is based on binaries, on code and things being and or and not, and things being very clearly defined one way or the other. And you have also previously talked about how you view DNA as akin to computer hardware and the physiology of living cells more as the software, if we're mapping organisms onto computers for this analogy. So how do you reconcile the inherent messiness of nature with what you've often described as the idea that nature is programmable, because we have this hardware and software that we are now learning how to play with?
Yeah, well, I think there are a number of important ideas there. One really important aspect of Xenobots and technologies like the Xenobots, meaning chimeric types of technologies, various things that others are doing as well, are really asking us to take a deeper look at terminology that we've taken for granted. So it used to be that terms like robot, machine, and so on, it was very easy to tell that apart from living things because living things arose via an evolutionary process. Robots come off a factory, robots are metallic, they're boring, they are predictable, and so on. These technologies are reminding us that those were never really good categories to begin with. They were okay when we were dealing with the limitations of robotics and the materials that we had, and in the biological case, the sort of n=1 example of the way that life progressed through the tree of phylogeny on Earth. And being able to make these kind of novel, so all of the bioengineering, the synthetic morphology, all of this kind of work reminds us that these categories are much softer than we thought they were, and we are going to have to look for deeper understanding of what it is to be a robot or a machine that doesn't rely on these very contingent facts of what's it made of and how did it get here, right? And so for where the hardware and software comes in is in the amazing fact that the Xenobots, like other examples in biology, what they're telling us is that evolution seem more than just generating specific solutions to specific environmental problems. What the genome of any organism really encodes is a kind of process that can identify novel ways to be a coherent organism in novel circumstances. They can solve problems which they may never have seen during the evolutionary process. You know, these Xenobots are not a normal part of life of the frog or its ancestors, as far as we know. And so while the genome nails down the hardware that every cell gets to have, right? So the genome specifies the proteins that every cell is going to have, the actual outcome of what collectives made of those cells are going to do in specific environments and manipulated in specific ways, as directed, for example, by this machine learning algorithm. The outcome of that is the result of all kinds of cell-to-cell communications, competition, cooperation, all of these interesting aspects of a collective intelligence that solves problems as other people have been studying in ant colonies and in swarm robotics and in lots of other contexts. So that's I think that's the important distinction here. It's not that these things are like the kind of computer with which most of us are familiar. That's not the point. The point is it's reprogrammable in the sense that the hardware spec that's produced by the DNA does not once and for all nail down everything that a cell is capable of. So cells with a perfectly normal frog genome, yes, by default, they make tadpoles and frogs, but actually they're capable of doing all kinds of other interesting things. And that's part of understanding the software of life, is to get a better handle using this, very tractable, very, very self-contained, safe platform, is to use it to understand what are these active subunits like cells, like swarm robotics, like the Internet of Things. What are these things capable of in collections? Because we are all confronting them in our life and in other aspects of life.
Yeah, awesome. That's great. I love how you talked about the fact that the hardware and the software and how those terms actually do really map very well to what we're trying to talk about here with how trying to understand these living organisms and sort of some of the advantages, I think, of sort of existing what you call traditional robots or things like hardware and software is that we know how they work. We made them. People talk about code being a black box when it comes to artificial neural networks. But I think in general, that we kind of have a system for how code is written and how these systems are built. But when we're trying to understand how nature works, there wasn't really a blueprint. It was a lot of trial and error. And as Mike just pointed out, you might have one set of genes, but you can build so many different things in so many different ways with those genes. And how does that happen? And how is that modulated? And so my next question is for Josh, kind of bringing us back to the computational side of things. I would say that the fact that we have been able to build things like robots and hardware and software have allowed us has given us kind of a vocabulary and a way of understanding life, to the point where a lot of people are trying to build simulations of the human brain out of computers, and sort of thinking about the brain as a collection of circuits, I think, has really been a big revolution in how people are now trying to tackle that problem. But I'm curious to hear from your perspective the inverse. How can using things like Xenobots to understand biology help to inform our design of future systems and robots, and maybe even computational platforms and AI?
Yeah, that's a great question. The way to sort of draw inspiration from Xenobots in particular and biology in general is, again, to compare and contrast. So if we view the Xenobots as robots and then look at our current state-of-the-art robots, we can ask, how do they act differently? Someone on the panel mentioned that robots are not what they're made from, but what they do. Xenobots act very differently from traditional robots. One very clear difference is current state-of-the-art robots and AI is very easily fooled. There was a great recent example from a few weeks back in which somebody wrote the word iPod on a piece of fruit and presented that piece of fruit to an AI. And the AI was convinced beyond a shadow of a doubt that it was looking at an iPod rather than a piece of fruit. It's kind of a humorous example, but it sort of points out one of the potential limitations, the potential danger of our current AI technology. I think evil AI like Skynet may be scary, but what's even scarier is an increasingly intelligent entity that makes mistakes under certain conditions and doesn't know that it's made mistakes. In contrast, if you look at the Xenobots, they can't be fooled so easily. Doug did a great experiment in one of our earlier papers where even if something surprising happens to the Xenobot, like being cut almost in half, the injury is not surprising to the cells that make up the Xenobot. The cells somehow talk to one another and figure out how to stitch the bot back together again. So that tells us that in AI and robotics, we need to try and make machines built of machines built of machines. Individual cells are themselves more sophisticated sensor motor coordinated machines or robots that are beyond the capability of any existing robots. So if you build machines of machines of machines, then it's very unlikely that the world is going to be able to surprise all levels of this multi-level machine at the same time. That's something that I've learned as a roboticist from working with Doug and Mike on the Xenobots. And I think it suggests how Xenobots might blaze a new trail for us towards trying to achieve both useful and safe, or not easily fooled, AI. Next piece of this is, at the moment, we've been sort of using the AI to design Xenobots from an engineering point of view. Get the Xenobots to walk, get the Xenobots to swim, get the Xenobots to replicate. One thing we're looking at doing in future is trying to tell the AI to design a Xenobot for us that's conceptually transparent. That as the Xenobot goes about and does whatever it does, that it advertises to us its form, its internal function, and its adaptive behavior. And if we can see all of those three things, form, internal dynamics, and behavior, happening at the same time, we might be able to pull out increasingly subtle mechanisms from biology and figure out how to build those into useful and safe AI and robots.
That would be so cool. Just to watch a Xenobot yeah, just watching a Xenobot doing things and have it tell you, now I'm doing this because I found this thing, or I hit this, I hit what I think is the edge of the dish, so now I'm turning around. That would be so cool. So I have one more question for the panel, and then we will get to the questions that our audience has been asking for the last 36 minutes. They are great. Some of them are very specific. Some of them are very general. So I'm excited to get to those. But my last question is going to be the same question that I'm going to ask all four of you, and I'm going to let you each answer individually. This is the third paper that your team has published about the Xenobots. As Josh hinted at, the first paper was published in 2020, only a year ago. It's hard to believe. What is time? You created the Xenobots, and then in the second paper, you showed that they could self-assemble from individual cells and that they lived longer and that they could record information. Now they can replicate. So my question for everybody is, what is next for this team and for this project? And what aspect are you most excited about? And that can either be in the immediate, what insights are you hopeful the Xenobots will reveal to us, or it can be down the line, what could these things possibly be used for, other than just really cool delving into the roots of biology. So I'm going to just go through the alphabet by last name. So Doug, we'll start with you.
Sure. There's two things I'm interested in. One is very basic, which is we're really sort of working with a very small set of a Lego set, right? If you think of all cells as different Legos, we have a number of frog cells right now, but the possibility of the different types of cells that we could use to examine a system like this is huge. That space is quite big. And so I would love to see this move to other types of organisms, other cell types, and other environments, and sort of see the way that we could design and learn about how these systems would behave. And then on the more practical side, I really like this idea of sort of understanding how to generate or modulate goal-driven behavior. So what we see now is a lot of sort of spontaneous motion and emergent behaviors. And so if we could start to program or dictate the way that these move and sense stimuli and respond to stimuli, then you can start to get some really interesting programmable behaviors or guidable behaviors. And we can maybe talk down the line as a group about how that would certainly be really useful for biomedical and environmental applications as we move forward.
Great. Thanks. Josh, you're next.
Yeah, I sort of answered your question already, I guess. As an AI researcher, I'm most interested in figuring out how we can whisper in the ear of the AI to get it to design new kinds of living systems. We've seen recently AI designed proteins, AI designed drugs. Our group is tackling AI designed biology and organisms. How do we get creative AI to make useful living systems that do what we want and don't do what we don't want them to do? I think this is an open challenge that's going to involve many more researchers on the AI and biology side. You're muted, Lindsey.
Wow, sorry. I got my alphabet mixed up and thought it was Mike, but it's Sam. K comes before L. Go ahead, Sam.
Okay. So there are several motivations for this work, as we mentioned, spanning robotics and biology, and there's several different directions this work could go. We can only maybe go down a few of them, but there are scientific trajectories of interest, like Josh mentioned, learning more about using AI to make the Xenobots advertise their underlying mechanisms so we can learn more about developmental biology. There are philosophical lines of inquiry that we can follow. What is it like to be a Xenobot? Probably nothing right now. It's like a skin biopsy, probably nothing more. If we start adding more sensory organs that affect their actions, as Doug was mentioning, at what point does this become intelligent as a tadpole? I don't know. Those are interesting philosophical questions. As a roboticist, I really would like to somehow create useful technologies with this Xenobot platform. And in robotics, we've been very good at making robots inside of highly controlled factories where everything is exactly where it needs to be. Temperature, humidity, lighting is always very, very highly controlled, and the machines are made to be super precise. And we're very good at making those machines, and they've revolutionized those industries such as car manufacture. Robots build our cars, but they don't build our skyscrapers. And there's a key difference there because skyscrapers are built in the outside where there's all kinds of unpredictable conditions in the outside world. And it's been a major challenge in robotics to try to create robots that are useful in the wild, where you can't predict what's going to happen, where there's surprising conditions, as Josh mentioned. I don't know what the Xenobots are going to be able to do in the future. Right now, they're exceedingly simple, but they have a lot of properties that are inspiring. And one of them is natural. It is not inspiring. They're made of cells, or it's not surprising. They're made of cells so they can biodegrade. So if we can control this system to do something useful, then that suggests that it would be able to do something useful in the environment without adding more pollution. It would just biodegrade, gracefully disappear. And that's something that's very exciting. And I hope that we can find a useful application in environmental cleanup jobs, for example, in the near or mid future, sometime within my career.
Awesome, thanks. And we're going to end with Mike.
I want to shine a light a little bit on the implications of these kinds of technologies for a really pressing need in the human medical and veterinary world, which is the incredible amount of biomedical suffering that's out there. And it's kind of interesting to think that with the exception of infectious disease, most medical problems, and we're talking about birth defects, traumatic injury, aging, cancer, degenerative disease, all of these things could be addressed in a really radical way, much more definitively than is being addressed now, if we had the answer to one simple question, which is, how do you get collectives of cells to build a specific thing that you want them to build, right? And the real ability to do this, the ability to control the outcomes of anatomy, of structure and function in cellular collectives, is not going to be solved alone by stem cell biology or by genomics. It's going to require not only understanding the molecular mechanisms that are required for interesting things to happen, but also the algorithms that are sufficient for them to happen. In other words, the kinds of information processing that are required for this collective of intelligence of cells to build a complex structure, to build a different structure under perturbations or in a new environment, to repair that structure. We really need to understand the kind of intelligence of this process in the sense of collective intelligence, right? Not in a magical sense, but in the way that it's being studied by lots of people in that field. And what the Xenobots to me represent are a really powerful platform to begin to ask that question and to crack that code leading from the stimuli that you give to particular cells to the final outcome. What is it that they make, so that we can learn to make all kinds of things. And the end result of this, I think, down the line, years later, the end result is going to be a completely different sort of regenerative medicine, where we can really address the need for complex organs that we are not able to micromanage from the ground up. We're not going to be able to anytime soon to be able to build a hand or something like that from individual cells directly. But we may be able to motivate or convince a set of cells to build it the way that it did once during embryogenesis. So that to me is really the critical aspect of this technology. It's that the understanding of how it is that you control the anatomical and the structural outcomes of cellular collectives.
Cool, thank you. So I'm going to now move us into questions from the audience. A lot of them have been sort of addressed as we've gone through the conversation, but one of them that is kind of building off, Mike, what you were just talking about for regenerative medicine. Sam talked about how it might be possible to use the Xenobots in kind of a future iteration of them as a collective to achieve things like going out in the environment and remediating, sort of performing tasks. But when we're talking about things that would be happening in medicine in the human body, I'm imagining, and just want to clarify, if you can clarify if this is true, that the Xenobots themselves would not be sort of used for that, but they would generate the insights that we need to then be able to go to a patient and deliver the right messages to their own cells so that the patient's body would do the thing that we want it to do. But the Xenobots themselves, we wouldn't basically use them like the proverbial nanobots that you inject into someone's body and they build stuff. Is that correct?
Well, I actually think both of those are viable paths for future progress. So I think we can certainly imagine, once we get a better handle on creating very specific kinds of bots with specific functions, you can certainly imagine them either micro-sculpting some sort of tissue in vitro for then transplantation into patients, or possibly used directly in patients at some point to chase down tumor cells or interact with bacteria in the gut, or to clean out arthritic knee joints, or to put down some sort of pro-regenerative molecules. I think it's definitely a possibility that these things are used directly in medicine, but I think the longer term future is the other thing that you said, which is to use them to understand how it is that we get cells of the body to execute repair, directly.
Cool. I'm sure that a lot of people would like to be able to, just grow a new spleen, grow a new kidney. That would certainly solve the organ transplant problem we currently have. Another question from the audience. I'm going to give this one to Doug because it's about the cells that the Xenobots are built from. One person in the audience pointed out that stem cells do differentiate. They usually don't stay stem cells forever. They become eyeballs or lungs or whatever. So do the stem cells that make up the Xenobots eventually differentiate into something? And if not, then I think you mentioned 10 to 14 days is when the cells kind of fall apart. But is that because the stem cells are developing and getting out of sync, or do the stem cells not differentiate in the Xenobots?
Yes, so there's two separate answers to this. The stem cells that we use as the feedstock. So if you place loose stem cells in the dish, those go through some small level of differentiation and then die off. They're missing a lot of the signals that are necessary from their immediate environment and their neighbors to form some sort of complex structure, differentiate effectively. The ones that make up the Xenobots, it depends on how the construction method you're talking about. So some of these have muscle tissue, some of them have skin cells. Those are pretty well characterized. So it's known the developmental trajectory. This is work that's been done now for 20 years that leads you from being an undifferentiated cell that has a fate to become something on the outside of the body into becoming one of the 4 or 5 known cell types that make up frog skin or tadpole skin. And so that process happens automatically. That's a built-in feature of the system that, as long as you keep the physiological conditions correct and keep the water correct, this is an emergent property that happens over about 3 or 4 days of development. Now, that can be pushed and pulled pretty effectively. We have molecular reagents, pharmacological reagents. You can even use mechanical strain to physically push and pull on the cells, and that can alter the decisions that are made along this trajectory. And those are the types of tools that we could use if we wanted a more top-down approach to dictate specific types of interactions between the cells.
Great, thank you. Next question from the audience is for Sam. Can the Xenobots self-replicate without human intervention? The videos indicate that they had to be first altered by cutting out a small piece physically to make the little Pac-Man mouth shape. So if the replication that we saw with the Pac-Man shape is not possible without the human interaction of cutting the Xenobots into that shape, can we really call it self-replication?
Yeah, that's a good question. So no, there's still human intervention here. If Doug doesn't feed each generation with more loose stem cells from which to make piles that become offspring, then the system stops, replication terminates. That could be done by a machine, ostensibly, but here it's Doug, so that's human intervention. I think that it might be a mistake to think that self-replication is not self-driven if it relies on the environment. And all replication relies on materials, and the environment is just which materials they are and how small they are and what they are. In this case, the materials are cells. We don't go down any further than that. And they happen to be supplied by Doug each generation, but I believe that could be automated. But there is another part where the human comes in. I designed the AI program that was a human intervention, but these human interventions are minimal and much of the process is automated, which is very interesting from a scientific perspective.
In some ways, again, that is reassuring that you still need to have a human involved. We're not going to have runaway Xenobots anytime soon. Next audience question is for Josh. What level of complexity will Xenobots achieve? At what point do you think you will need a nervous system? And do you have plans of connecting the Xenobots to brain organoids or other neuronal tissue? So kind of combining the biology and the computation there.
Yep, that's a great question. You can probably guess that's on our to-do list somewhere, a list that grows ever longer. And I think this, again, relates back to trying to get the AI to design Xenobots that clarify the biological dark matter, the things we don't understand very well. What are the kinds of things what kinds of tasks could we set for our Xenobots in which they're eventually going to need brains? So we could obviously put neurons in and see what happens, but alternatively, we could pose a number of challenges to them and see which ones are tackleable by aneural Xenobots, those lacking brains, and which tasks are tackleable by neural Xenobots. And if we were to do those hypothetical experiments, that would give us unique insight into the evolutionary pressures that favor nervous tissue, the centralization of sensation and action. We have ideas, we have hypotheses, but again, these are things we sort of dreamed up in a vacuum. It would be great to see if we could design Xenobots that tell us the conditions where brains are actually required and bodies alone are no longer sufficient.
Well, thanks. My next question is for Mike. This person says that they have been following your work on non-neural bioelectricity, and they're wondering if it's possible to bioelectrically manipulate the Xenobots to get them to assume a certain shape. In the current study, all of them were hand sculpted basically by Doug, but could you use bioelectricity to inform how they are shaped?
Yeah, that's a great question. That is absolutely on our list. We in the future are going to be testing all sorts of ways to manipulate the conversations that these cells are having with each other to that result in specific structures and functions. And as you say, many of those conversations are electrical in nature. And so I think absolutely there's going to be a really important place in tracking and modifying the electrical information processing in these cells to achieve specific programmable outcomes. Yeah.
Cool. Next question, I'm going to make it a popcorn question because I'm not sure who is the most equipped to answer it. One person is wondering, are there any specific biological design principles that you all have identified through your work with Xenobots?
I mean, I guess I'll take a, go ahead, Doug. Okay.
I was just going to say, I think what's interesting, and for me, so I have no robotics background, and I think what's interesting for me is I was completely ignorant of this field just with my own training, and there are such interesting design principles from these other fields, from artificial intelligence, from robotics, that have not effectively been integrated into biology yet. And so we typically think about biological design from the point of view of what we see every day, which is organoids. So you start with a cell or an explant and you regrow an organ. But this idea of, rather than a single cell that's synthetic, what if you could build from the ground up, from any possible shape, any possible design, what would you build? How would that behave? I think this is a different way to think about assembling multicellular agents, and that developmental techniques have not been brought to bear on this question in any meaningful way. And so what we're seeing is that these fields, it's really nice to start to have a blender of these techniques. And it's not really about what is this, what is that, what is this called, how is this used? It's really these different tools for the jobs and different ways to interrogate a particular question. And so it's been really fun for me to learn about design principles from these other fields and sort of bring them into the realm of biology and start applying them to these questions.
Great, thank you.
I would say, just to build on Doug's question, from the opposite side, being somewhat naive about biology, I realize now in retrospect, we're building robots out of parts that have been around for 3.5 billion years, and they've been attending the school of hard knocks for all of that time. So you can rearrange them, you can do all sorts of things to them, and they somehow manage to find a way to form into some stable, higher level structure. That is completely missing from all of our technology: robotics, AI, computers, vehicles. You rearrange the parts in your car or your phone, and you have an inoperative machine. I think that is sort of the one meta biological organizational principle that I've taken away from this work so far.
Yeah, and building on both of those concepts is this idea that what we have in biology is this kind of multi-scale competency, where every level of organization is, everything is not simply made of parts, but each of those parts is competent in solving important problems in various spaces. And it remains to be understood how, taken together, that kind of architecture gives this amazing plasticity, where these cells that have evolved for eons to have a very particular reliable behavior and turn into a tadpole and so on, under novel circumstances, they will figure out a way to, for example, self-replicate in an entirely novel way that they never had to do before. So this ability to solve new problems in new environments in new ways is somehow an outcome of the evolution tinkering with this multi-scale architecture. And that I think is very powerful on both ends. We have to understand it both in biology and on the computer science side.
Cool, thank you, everybody. And one thing that I love about this team is the different expertises that you all have, and how, as you've mentioned, you've kind of come together to bring the biological and the robotics and the computational backgrounds together to make these things that I'm sure that anyone on any one field, any one discipline probably could never have come up with. But here they are. We've seen them. They're tootling around dishes, and now they're replicating. And I'm excited to see what they're going to do in the future. So I'm going to invite Josh Brown back on to close out the session. And we had so many great questions that we unfortunately weren't able to answer live on air. We will try to answer them in the days and weeks to come. If you want to put your questions on the Wyss Institute's YouTube channel, we are going to be posting a recording of this video. So if you have specific questions, you can leave them as comments on our YouTube page, and we will do our best to get answers to them. So let's go ahead and turn it over to Josh. Other Josh. There are two Joshes on the panel. It's confusing.
I like to think of myself as Josh the Lesser. Anyways, thank you all so much for this magnificent and insightful conversation. Especially thank you to Lindsey and the whole team there at the Wyss Institute at Harvard for this terrific work. And there's so many people behind the scene that pull science together and pull events like this together. So thank you to you folks. And thank you to this science team. It's a remarkable a line of work you're involved in, a remarkable team at the University of Vermont, at Tufts University, at Harvard University, working together to make this this set of insights and this path into a really remarkable future. So thank you all for this. You've been listening to conversation about Xenobots, and the study appears in the Proceedings of the National Academy of Sciences. It's called Kinematic Self-Replication in Reconfigurable Organisms. Thanks all for listening in. And as Lindsey said, in the weeks and days ahead, we will attempt to answer the various questions that are coming in from around the world. So thanks all for your
Thank you so much.
wonderful insight.
Thank you, everybody.
Thank you, everyone.
Bye.